Okay, so we will start today's session. So, a very good evening, everybody. So, let's start. Okay, so in today's, am I audible?
Yes, sir. Yes, ma'am.
Okay, fine. So, today we are going to discuss the portion covered in the last week, that is isolation and purification of products. Okay, so in this particular lecture, we are going to see the downstream processing which is involved in the processing of any product. So up till now what we have studied that suppose we want to make a drug. Okay, we have made a genetically modified, so we are studying genetic engineering. So we have studied up till now how to create a genetically modified, uh, not an organism, but how to have a genetically modified cell, and how to introduce a foreign insert into a vector, and then how to express it so that we can have the protein products. And now that we have our protein products in our hand after cloning it and then expressing it using expression vector that we have studied in our last class, the next thing is that now we want to recover those products. Okay, so up till cloning and expression, whatever things that we do, that is known as upstream processing. Okay, and after the expression of the product, suppose if we have a protein product, after the expression of that product, whatever all we do, that is known as downstream processing. Fine. So downstream processing means that if I am talking about the production of a particular protein, a recombinant protein that I have produced, so the recombinant protein I have produced through genetic engineering. Okay, so I have produced a recombinant product.
Ma'am, your screen is not visible.
My screen is not visible. Oh, okay, okay. I'm so sorry. Let me just share my screen. Now it will be visible. Hmm. I think now it will be visible. Is it visible now?
Yes, ma'am.
Okay. So,
Yes, ma'am.
okay. So now we have produced a recombinant product. Fine. So this recombinant product we have produced how? So in the first stage, what we did, that we amplified the gene of interest from the genomic sequence, and then we took a vector with the same restriction enzyme. We had digested our insert as well as the vector. We ligated it, we transformed it, we screened it, and we got the product. This is how we cloned it. And suppose I want to express that particular gene also, then I have used expression vector instead of cloning vector, and I have put some extra more sequences like initiation sequence, termination sequence, ribosome binding site, etc. And I have expressed the product. Now the protein product is there with me. Now my next task is to isolate the product. Okay, so for the isolation of product, what all steps that I'll be doing, that is known as downstream processing. Fine. So up till the cloning and expression, the steps, the whatever things that we do, that is known as upstream processing. And the purification and the isolation and purification and recovery of the product, that is known as downstream processing. In fact, whenever you are doing any kind of drug discovery, or suppose in large scale when you do the production of any kind of drugs or any kind of recombinant product, suppose we have recombinant insulin and many more recombinant products are there. So actually the major cost of production is dependent on the downstream processing. So it's the downstream processing that determines the cost of any particular drug, any particular recombinant product, because the most expensive part of the whole thing is the downstream processing thing. Okay, so if we just we just let's just assume that what all we have to do when you have to do the downstream processing things. Suppose in a cell I have my recombinant protein. So what is the first thing that I have to do? First of all, I have to disrupt the cell, I have to lysis the cell. Whenever I will lyse the cell, then only I will have everything that is inside the cell, and that has my protein of interest also. So suppose the first thing is that I have to disrupt the cell, I have to lyse the cell. Okay, so whenever I will lyse the cell, there can be two options. The first option is that, just a minute. So for downstream processing, the first thing that you have to do is cell lysis or cell disruption. Okay, there are various methods of cell lysis and cell disruption. You can have physical methods of cell lysis and cell depression, you can use chemical methods, you can use enzymatic methods. So many methods are there depending upon the context and depending upon the type of cell that you have. You will choose your cell lysis or cell disruption method. Now this is my cell, this is my cell, I have lysed it. Now everything is there, each and everything is there. Okay, next step is that whatever my product is, it can either be intracellular or it can be extracellular. Okay, so if it is extracellular, then the need of cell lysis and cell disruption is not needed. But if it is intracellular, that is within the cell, then your cell lysis or cell disruption is very much needed. And suppose uh, I mean, it is not very much the case here. So okay, let's just assume that it is your intracellular, then only you are doing the cell lysis or cell disruption thing. Now I have my protein product with me. Now what I will do is that I will try to recover it. Okay, so for recovery we have many things. Then I will try to purify it, and after purification I will try to concentrate it. Okay, so there are many different ways, many different methods of purification, of recovering, of concentration. But in general, you just have to think that you have a cell. First thing, if the product is intracellular, you have to lyse it so that everything that is inside it comes out. Now, since there are many things, so you have to recover your protein of interest, you have to purify the particular thing that you want, and then you have to concentrate it, and then again, further purification or whatever steps are there that you have to do. So, in accordance to that, what we have studied in our first lecture is protein recovery from host cell. That means what are the different steps that are needed in protein processing. So if we just look at the different steps, so your product can be either extracellular product or it can be an intracellular product. If your product is extracellular, then there is no need of cell disruption methods. But if your product is extracellular, then you have to intracellular, then you have to do the cell disruption method. After cell disruption method, you have your crude product. Now you will extract means that you will, whatever crude product is there, you will extract it, you will concentrate it or purify it. See, they here I told you first purification concentration, it depends on the kind of the product that you are isolating. And then finally when you have the product, then you can use it for whatever purpose you have to use it. So but this is like the general uh, the kind of general protocol, which not a protocol but a general kind of flow chart that is involved in the downstream processing events. So now first we are talking about the cell disruption methods. Now cells can be disrupted with the help of physical methods, with the help of chemical methods, or with the help of enzymatic methods or mechanical methods. Okay, so if we talk about the physical methods, they can be your thermolysis. Thermolysis, as the name suggests, lysis means lysing, thermo means heat. That means that here you are using heat for the lysis of the cell. Okay, and since you are using heat for the lysis of the cell, then it is applicable to only those products that are thermostable. Suppose I have a recombinant product which is very thermolabile. Thermolabile means which is very temperature sensitive. So I cannot use thermolysis because it can damage my recombinant product also. So that is what I said, that whatever method that you have to use, there are n number of method. It depends on the kind of product that you have produced and what are the downstream applications of it. So thermolysis method can be used only when your product is thermostable. Second method that we can use are osmotic shock method, where when you are exposing your cell to a hypotonic solution, then what is happening, that your cell is getting swelled and ultimately it will burst, and when it will burst it will release the products. So you can use the osmotic shock method. Then you can have sonication methods. So in sonication methods, what do? You give ultrasound waves of more than 20 kilohertz, and therefore there will be so much compression and decompression events that whatever the plasma membrane or cell wall is there, it will get lysed, and then your all the products will get out of it. That is your sonication. And then you have so many chemical and enzymatic methods, such as alkali treatment you can give. You can have detergents, you can permeabilize your cell wall, and then you can have enzymatic digestions also. So enzymatic digestion means that you can use enzymes that can digest the cell wall, the cell membrane. So you can use lysozymes, you can use pectinases. So like this, you can use many kinds of enzymes that can uh, uh, damage or lyse the cell wall, cell membrane. Then you have mechanical methods like pestle and mortar that we all use in our day to day lives in our kitchens. And then we have many different methods such as bead mill disruption, high pressure homogenizer. So many methods are there, but the ultimate goal of all the method is that if your product is intracellular, then you have to disrupt the cell wall, or if or the cell membrane in animal cells, and you have to release everything that is present inside the cell. Okay, so that is what we have studied in our first lecture, that we have to disrupt the cell, and then we have to get recover of each and every thing that is present inside the cell. Fine. So now we'll do the questions of this particular just a minute. Yes, Naraini, you have raised your hand.
Um, yeah, ma'am. So yesterday we did a cell lysing method in our cell biology practical, where we used a plate, a bacterial plate, which we previously created.
Okay.
So I had this question because we didn't know which kind of bacteria we had on the plate, but we still lysed it. So how can you be sure that after lysing a particular like bacteria which you have got, how can you be sure that this is E. coli or it's any other type of bacteria?
First
So
of
how can we
all,
find that?
see, first of all, this basic information, you should always know that whatever you are doing, what experiment, whatever organisms, whatever bacteria you are using, first thing. But if you don't have the knowledge, then there are many specific tests which are related to strain specific tests are there. So you can search. I don't remember the exact test that can be used, but there are so many strain specific
No,
tests.
because the plates which we used were from a previous like environmental biotech practical
Yeah,
where we
because
had the
see,
soil sample.
I
So
understand, because you are doing some masters. You are in bachelor's or in masters.
I'm in second year BSc.
Huh. So because you are just doing some bachelor level of experiments where you just need to have like a basic knowledge or an exposure of how to lyse a bacterial cell. You don't really need that much of information of behind and of what has been done or what has to be done after that or what has been done previously. But when you are actually doing an experiment, then obviously you will have the knowledge of what you are lysing, right? But even if you don't have the knowledge, then you can always, first of all, you can see that so like what kind of plate you had. So it was, it a normal agar plate or it has some antibiotic resistant plate, was that, do you have any information like that? Was it an ampicillin resistant plate or any
It was a
chloramphenicol?
normal agar plate. It was a normal agar plate, and we had a one soil sample which one our lab had, and we
Huh.
growed
So then
grew it.
then you have to do some strain specific tests. So there are different strain specific tests for gram positive and gram negative bacteria to identify whether the bacteria that you have are gram positive or gram negative. Then amongst the gram positive and gram negative, the different kinds of strains, you have different strain specific tests. I don't remember the exact names of all the specific tests, but suppose X kind of bacteria you have used and you have lysed. So see, in general, what how to identify a particular bacteria or any particular strain or any particular thing is, it depends on the unique feature that is present on that bacteria. Okay, so whatever unique feature that is present in that particular bacteria and in that particular vector, in that particular plasmid, and this unique feature concept applies to everything, not just to identify that in your case the kind of bacteria that you have used, but it also depends, like for example, if you are doing some high level of mammalian research also, in that context also it depends that whatever unique feature is present, that you can exploit to to identify that particular bacteria. First thing. Second thing is, suppose you have n number of bacteria, okay, and you don't know that which what all bacteria are present, but at least you will have the knowledge of the bacteria of interest that you want. Suppose if there are 5 gram positive bacteria present in your plate, but you know that which gram positive bacteria is my bacteria of interest that I want to study, then you can use the test of that particular strain specific, and you will see whether your results are coming positive or not. Just like if you remember, uh, so have you done in 11th and 12th, have you done salt analysis, where in salt analysis you have to identify the salt. So you have a particular, like you have a basic chronology, that first you will test this, and then you will negate, and then you will subsequently move to other kind of tests. Have you done salt analysis in your 11th to 12th? You must have done that. I think everybody does that.
No,
You
no,
have done.
actually, we were COVID batch, so no.
Okay, but yeah, I mean, if you have even studied about the salt analysis, so it's like that. First you test the broader spectrum, and from the broader spectrum you see what all positive results you are getting, and from that broader spectrum you eventually try to narrow down your tests. So that is a way of identifying any particular thing, which is not unique, but with the information of which you don't know. From the broader spectrum you try to come to the narrow spectrum. It's always the way of identifying something which is unknown. Okay. Anyways, so now we'll move to the questions. We'll start the questions of our first lecture, which was the protein recovery from host cell. So I think I have already discussed a lot of things that I have made questions of. Anyways, so the first question is, if the protein product is thermostable, which is the method of choice for the cell disruption? Osmotic shock, thermolysis, sonication, alkali methods. What is the answer? If the product is thermostable. We have just discussed that if
Thermolysis.
yes, thermolysis. So thermolysis, as the name suggests, lysis means lysing, thermo means with the help of heat. So that means you are lysing the cell wall or lysing the cell with the help of heat. But you can only use this method if the product is thermostable, otherwise your product may get denatured, it may get damaged. So if the product is thermostable, you can go for thermolysis method. But it's not like that you have to go for thermolysis method only because my protein product is thermostable. No, it depends on many other factors. But the basic criteria, thermolysis can be used only when your product is thermostable. But thermostable product can be used, can be purified or disrupted by other methods also, if you are getting better recovery from any other method. Now, next question is, Rajesh has expressed a protein using Agrobacterium tumefaciens plasmid. Okay. However, while performing the osmotic shock for disrupting the cell, he could not recover the product. What can be the possible reason for it? The solution used for osmotic shock was hypertonic, therefore the cell could not burst. Plant cell cannot be lysed by osmotic shock method. Cell membrane could not break as he did not perform the experiment carefully, or none of the above. What do you think can be the answer? Think. Try to think logically what can be the answer.
Option B.
Uh, just a minute. I have to just check one thing. Yeah. Yeah. So you're saying option B. Option B is plant cell cannot be lysed. Yes. So option B is the correct answer. Why? See, this thing we have learned, we have just studied, or if you have studied the lecture, that osmotic shock method is better when you have a cell membrane. Cell wall is very hard and thick, so osmotic shock method is not the method of choice when you have to use a plant cell. Okay, this statement is correct, but how to relate this statement with the question? Because here the plasmid that they have used is agrobacterium tumefaciens plasmid. Okay, here in the question nowhere they have written that they are doing it on the plant cell, nowhere they have written, but they have given a hint that the plasmid that they have used is agrobacterium tumefaciens plasmid. And we have studied in our previous classes that agrobacterium tumefaciens plasmid is used in plant cells. Okay, so that means that he is dealing with plant cells, but he is performing osmotic shock method. But osmotic shock method does not work well when the cell wall is present. That means plant cells cannot be lysed by osmotic shock. That is the answer. Is it clear to everybody? Agrobacterium tumefaciens is a vector which is mostly used for plant cells. So that means he is performing osmotic shock methods in the plant cells, but plant cells cannot be lysed by osmotic shock method. That is why he is unable to recover the product. Just a minute. Okay, someone has joined. Fine. Okay. Uh, yeah. So next question is, continuous compression and decompression is a result of high electric waves sent to the fluid, osmotic shock, giving high amount of external heat energy, or ultrasound waves. So continuous compression and decompression, where does it happens? So continuous compression and decompression is a property of which kind of method?
Sonication.
Sonication. So sonication involves continuous compression and decompression. So if we are dealing with sonication, that means what we are dealing? Osmotic shock, no. Heat energy, no. Electrical waves, no. But there are ultrasound waves. So continuous compression and decompression is a result of ultrasound waves. Okay. Now the next question is match the following. So try to do it. Hmm. Try to solve this alkali method. What does alkali method do? Anything that you can give? Detergents you have studied. What detergents do? What kind of formations happens with the help of detergent? Basic. We have studied it in many times. We have studied in chemistry in class 12th, I assume. Yes, Khushbu is right. So detergent would be micelle formation. C. Okay, this is done. Now, bead mill disruptor. Bead mill disruptor. What can be the options from here? What do you think it can be? Bead mill disruptor. Saponification, titanium beads, toluene,
Titanium.
titanium beads. Yes, right. So bead mill disruptor will go to titanium beads D. Now we are left with alkali methods and permealization. So alkali methods. So alkali methods will lead to saponification of liquids, and permeabilization, it is toluene. We'll just quickly see this. This is lecture number 1. So alkali treatment, it causes lipid saponification, which disrupts the lipid packaging and affects the cell wall integrity. So basically, when you are giving alkali, so what will happen? The cell wall integrity is compromised because it causes the saponification of lipids. Detergents, we all know that it forms the micelles. I think this is something that we have been studying since class 11th or 12th in our chemistry. This was there. Permeabilization. So permeabilization is achieved with the help of an organic solvent, toluene, which was given in one of the options. Okay, and then next one is the bead mill disruptor. Bead mill disruptor is a mechanical method where, so where basically what you have, that the bead mill consists of a grinding cylinder fitted with a central shaft. So this is your shaft. What is shaft? This particular thing is known as shaft. Okay, this is your shaft. Now this shaft, you have to always remember. Why? Some things that you need to remember: this shaft, this impeller, this is your shaft which is rotating. These things, this is your impeller which is causing the mixing. Okay, now why I am saying to remember all these things is because I don't know whether you will have your bioprocessing subject or not, but if you have your bioprocessing subject, then repeatedly you will be using these terms: impeller, shaft. So the shaft is basically which is causing the rotation, and what is actually rotating are the impellers. And this jacket is there. So basically jacket is provided because wherever you are having a very large bioreactor, so what happens that because the large bioreactor is continuously moving and so much of product is inside the bioreactor, so there is lot of generation of heat. So that heat generation can affect the instrument and can affect the recovery of the product also. So therefore, to minimize heat, you have water jackets around the process, around the bioreactor. You have water jackets. So these jackets are actually the water jackets. Okay, and this is the inlet of the cell suspension, whatever you are giving, and this is the outlet. So that means you are giving your inlet because of that rotation of the impeller. This rotation is causing because of the shaft, your and because of the beads, whatever cell suspension is there, it is getting disrupted, and whatever product recovery that you are getting from the outlet. So it bead mill disruption means that the beads are helping in the disruption of your cell suspension. Fine. So someone is prompting, just a minute, just a minute. Okay, now in the next lecture 2 comes the basics of chromatography. Okay, so now we are studying about, here we today in today's class, whatever we are doing, we are studying about the downstream processing, downstream events that happens after the production of the recombinant protein. So I know that, I suppose I have an intracellular product, because for extracellular product, cell disruption is not needed. Okay, now I had my product is intracellular, so I disrupted the cell wall. So cell disruption I have done, completed, and now I have mixture of products, as in that inside the cell you don't only have your protein of interest or your product of interest. Inside the cell there will be so many things. Okay, so cell, after cell disruption, whatever output you are getting, whatever is your recovery, that is not just your product of interest, but that is a mixture of many different products. Now what you have to do is that from this mixture of so many different kinds of products, you have to isolate your product of interest. Okay, that means amongst the mixture, this is mixture. Okay, you have so many things here. From this mixture you have to, so other many different things are there. You have to separate your desired product. Okay, so how you will do it? So for this separation of the desired product, separation of your product of interest, we perform chromatography. Okay, now chromatography, what does chromatography means? So if we say chromatography, okay, so what is chromatography? Chroma means color and graph means kind of peaks. Okay, so initially, because of the difference of the colors that they used to get, they have different kinds of chromatogram peaks, but now it's more sophisticated. But this is how the name originally got originally got its name. So chromatography can be of many types, but here that we are going to discuss, that you must have studied in your lectures also, we will be dealing with column chromatography. We will be dealing with column chromatography. Okay, but before proceeding into this, first we will see, so in this class we have studied about the principle of separation, chromatography and chromatogram. So before proceeding to it, we have to see the basis of separation. Okay, I have two products, A and B, which are mixed together. So after the cell disruption, I got a mixture of A and B. Okay, now I want to separate A from B because I just want A. So now how A can be separated from B, or how any two products can be separated from each other? You can have some physical properties and you can have some chemical properties. Physical properties means they can have different boiling points, different melting points, they can have different density, and many other properties. These are the physical properties. Chemical properties means that they can have different functional group, or their reactivity towards different materials or towards different chemicals can be different. Suppose we have a chemical, say X, so B can have a greater reactivity to X as compared to A. So on this basis also they can be separated. So basically, to separate two different molecules, or to separate a mixture of molecules, we have to exploit some of their properties that makes them unique. Suppose I want to separate A from B, and I know that the boiling point of A is very less as compared to B. So what I will do, that this is a mixture, it has A and B both. It has A plus B. I will start boiling it. I know that A has a boiling point which is lesser than that of B. So first whatever is getting boiled, that is A. I will condense it and I will get A. So I mean, this is a very crude way of saying how to separate, but what I'm trying to emphasize is that the basis of separation is the difference in the properties, whether it can be physical properties or it can be chemical properties of the two components or the mixture of components that can be used or that can be exploited for separating them. So that is what they have also kind of, if you could see here, the principle of separation is the different properties. You can have physical properties, that means molecular weight, basic properties, molecular weight. If I want to separate A and B, if A has a higher molecular weight than B, then I can use that property, that is A, molecular weight, boiling point, freezing point, crystallization, solubility, density. So these are all your physical bases. These are all your physical properties. Apart from physical properties, as we just studied, functional groups, their reactivity towards other reagent to form complex. So two molecules can be separated on the basis of the different properties that the two molecules have. Now this is okay. This is like a basic principle of separation that we can use. Now we comes to chromatography. Now how to define how chromatography is exploited here? Just a minute. Okay, fine. So now how chromatography can be exploited is that here we are dealing about column chromatography. Okay, so the kind of chromatography that we will be discussing here is column chromatography. So column chromatography means, so you have studied here in the class, you have studied about ion exchange chromatography, size exclusion chromatography, or gel filtration, or affinity chromatography, or adsorption chromatography. So these all the kinds of chromatography techniques that you have studied or you will be studying, basically these all are column chromatography. So the column chromatography means that the chromatography is performed in a tube or in a column. Okay, that is column chromatography. And you have two kinds of phases. First is your stationary phase, and second is your mobile phase. So what does it mean is, I will just show you here what does column chromatography means. See, this is a column and you have a matrix, whatever matrix you have. Okay, or you can have some liquid also. It depends on the kind of chromatography you have. But essentially, whatever things that are inside the column, whatever things that are inside the column, I'm repeating it again, that kind of forms a stationary phase. And then what you give from outside to the column, it forms the mobile phase. Okay, so the mobile phase, because it is moving when you are putting this particular solvent, then it is moving, it is mobile. That is why it is called a mobile phase. It is called a stationary phase because it is fixed with inside the column. Okay, and then when you pour your this thing uh, mixture that you want to separate, the mixture will separate between the two phases. What I am saying, please try to understand the concept, because once this concept is clear to you, then whatever different kinds of chromatographic techniques that we will be discussing, whether it is ion exchange, whether it is adsorption, whether it is gel filtration, everything would be clear. But the basic thing, the basic principle is this, what I am trying to explain. And this is also very important in with respect to your competitive exams, whether it's a gate or CSIR or DBT, ICM, or any examination. They always ask a question related to related to chromatography, specifically color chromatography. Okay, so you have to understand the basics. If you do not if you could not understand, you can tell me, I can explain you again also. So what I'm trying to say is that mixture jo hai, it separates between the two phases. Okay, the two phases are stationary phase and mobile phase. Okay, and that we calculate with the help of their distribution coefficient, that is KD. That means how much the mixture has separated in the two phases. So that means if this is a column, inside you have a stationary phase and you give mobile phase from outside, and your mixture of proteins or whatever product is there. And if your product has two different proteins A and B, and this A has higher affinity for this particular matrix, then A will stick into the matrix. Okay, now since A stick to the matrix, B would be eluted first. So how the mixture has separated between the two phases? A it got bind to the stationary phase, and B, with the help of mobile phase, it got eluted first. So this is how the mixture, it separates between the two phases, that is the mobile phase and the stationary phase. Is it clear? The basic concept, is this clear to everybody? The basic principle
Yes,
of,
ma'am.
okay, fine, good. So the particular this but in this particular lecture, we have studied the principle of separation that I just discussed, that how the two different particular this thing how to separate the two particular products or mixture of products. Chromatography, now comes the chromatogram. Now this is also important, that okay, anything, see, one thing that you have to always understand is that whatever experiments you are doing in biology, whatever experiments you are doing in life science, in biotechnology or whatever, doing an experiment is one thing, and analyzing your results is another thing. Even if you're doing microscopy, even if you are seeing your images in a microscope, even then you have to analyze it, and then you have to produce the data in the form of a graph or whatever it is. So whenever you are doing a column chromatography, you have to represent your result in a way, and that representation is done with the help of a chromatogram, where you have different peaks. I'm sorry, the peaks that I made are not very good, but just for your illustration purpose. So the representation of the data, the representation of the chromatography is done through chromatogram. So chromatogram means it is the graph of the peaks that are observed during your chromatography experiment. And the chromatogram peaks, it will tell you that how efficiently your products have separated. How? Suppose I have two peaks, A and B. Okay. Then I have another two peaks, C and D. So these peaks are very well separated. That means each peak represents the molecule. So these two molecules have really well separated with each other. But these are overlapping peaks, infused peaks. These are overlapping. They are not very well separated. So these peaks are not very well separated. That means these molecules are not very well separated with the help of chromatography experiment. So a good chromatography experiment will give you distinct peaks like this. So if you are getting peaks like this, that means your whatever products were there, they have separated pretty well. But if you are getting overlapping peaks or infused peaks like this, that means your product has not separated, not this, like this. That means your product has not separated well. Okay, this is the basic thing. So here also you could see A and B. The peaks are so distinct. But C and D, these are fused peaks or overlapping peaks. That means these two are not very well separated from each other. So that is why a chromatogram actually describes the quality of your chromatography experiment that you have performed. Okay. Now we come to the resolution. So resolution means key whatever column you have. Okay. So resolution, whenever we talk about resolution, we're talking about the resolution of the column, right? So whatever column you have, that how that column is able to separate the two peaks is the resolution of the column. That means that just a minute, someone prompted. Okay, right. So that means whether your column is able to give you distinct peaks or not depends on the resolution of the column. So how you can see that how the resolution of the column is dependent? So it is dependent like if the column has a greater the length, greater the length, your better the resolution. Okay. And peak width. If it is broad, then your resolution is poor. Okay. So your resolution is directly dependent upon the length of the column, and it is inversely related to the width of the peak. So I have peak like this and I have a peak like this. It is a sharp peak. Okay. So this will give a better, this means it is a better resolved peak. So greater the length of the column, better would be the resolution, the less broad would be the peak, and more the width of the peak, the resolution would be very bad. Okay. So now we'll see the questions. The first question is, the principle of separation depends on differential concentration in the two phases, different boiling points, different molecular weights, none of the above, all of the above. What is the answer? What is the principle of separation depends upon? Tell the answer. Try to answer. All of the above. Yeah. So it is all of the above. So basically, whenever we are talking about the principle of separation, so if we are talking about column chromatography in general, but in particular if we're talking about column chromatography, then it would be just the differential concentration in the two phases. But in general, if we're talking about that how the separation basically depends, it depends upon the boiling point also, depends on the molecular weight also, and everything. So you can have the answer of all of the above, but if it is given the principle of separation during chromatography, then it should be differential concentration in the two phases, right? Now the next question is, to analyze whether the column has separated the two molecules properly or not, what parameters must be checked? Distance between the two peaks, average of base peak width, resolution, or all of the above?
B.
Just a minute, just a minute. Yeah, what you're saying?
B.
B. Okay. And anybody else who wants to try? So we have one option, B. Anybody else who wants to try? So the answer B is correct, and but here it should be all of the above. Why I tell you? Because you have to see whether the column has separated the two molecules properly or not. Okay. So you have to see the distance between the two peaks. Yes, because suppose if these are the two peaks, okay, like this, and these are the two peaks. So that means greater the distance between the two peaks means that they are that much far away. Matlab, they have resolved that particular. They have resolved even more properly, because these two are so nearby peaks that they are almost fused to each other. So that means these molecules are almost fused. So whenever you are trying to isolate this particular, suppose if it is A and it is B, so if you're trying to isolate A, just A, then there are so many chances that you can have your B molecule as well, because they are so much near to each other, they are almost overlapping. So although you have distinct peaks for A and B, okay, no problem in that. But experimentally, when you're trying to isolate just A because B is so near to A, then there are very high chances that along with A or B will also come. But suppose A and B, they have peaks like this. they are very distant, that means they are very properly resolved. Then when you are trying to isolate A, only A will come. So the two molecules separation also depends on the distance between the two peaks. Average of peak base peak width also, yes, that is but obvious true. Resolution, because resolution is directly proportional to length, resolution is inversely proportional to the width. So ultimately resolution is also the correct answer. So the answer would be all of the above. So this average peak with ultimately contribute to resolution only. Okay. So therefore the answer would be all of the above. Now, resolution during column chromatography is inversely proportional to length only, inversely proportional to length and height both, directly proportional to height, and directly proportional to length. What is the
D.
answer?
Ma'am, D.
Directly proportional to length. Yes. So resolution is directly proportional to length and it is inversely proportional to the width. Okay. Then next question is, during column chromatography, as n increases, peaks become more wide, peak width increases, resolution decreases, या peak width decreases. Try to answer. What can be the answer of this question? Just a minute. D. So Khushbu, are you saying D for this particular answer? I just saw your this thing message. Okay, peak width decreases. If you are saying, then yes, it is right. So during column chromatography, as n increases, peak width decreases. Now we'll just see how this n is related to the peak width and uh, the resolution. So here it is. Basically, if you have a column, how many distribution planes are there? That means how many times basically so it is beyond the scope of this particular class to explain you, but if you really want to have a even more better idea, like better idea, better understanding of how this works, there is a term which is known as theoretical plates. Okay, so basically what there was a problem. So the problem is that we know that as the length of the column increases, the resolution increases. Okay, this is something that we could understand from now. There is a message. Okay, just a minute. Someone wants to join. Fine. So just now we have studied that as the length of the column increases, resolution increases. So, but there is a limit to which we can increase the length of the column, because when you are increasing the length of the column, you are also increasing the time of illusion, which will have negative effect on ultimately the resolution. So there is a limit to which you can keep on increasing the length. You cannot keep on increasing the length up to infinity, up to any extent. You cannot do that. Okay, so just like in our cell organelles, so what they try to do is, you have studied that you in mitochondria and chloroplast, what they try to do, so they have folds, and these folds helps in increasing the surface area. Okay, so just like this, what we thought, what scientists thought, that okay, we cannot increase the height of the column up to any extent. So what we can do is that we will take a column and we will try to distribute this column, so that now with the same length we are distributing this column with the help of plates, so that now one time, two time, three time, four time, five time, six time. So now because there are so many plates, so there is it is with the same length, it is kind of increasing the resolution. Okay, so basically what they did was that they increased the distribution plane 1 2 3 4 5 by putting theoretical plates. So now as many distribution planes are there, that much your resolution will increase. So here they are saying the number of distribution plane in a column is controlling two parameters. First, as the number of distribution plane will go up, it will allow the analyte to travel for a longer period of time. Consequently, it will increase the distance between the two peaks. Okay. And as the number of distribution plane will go up, it will reduce the width of the base of the peak. As a result of, peak will be more sharp. That means distribution number of distribution planes is given by n, capital N. So as the n is increasing, your width is becoming even more sharp, your peak is becoming even more sharp. You can see when n is equals to 10, the peak is so broad. This is your n is equal to 10 peak. It is so broad. Kitna it has such a long width. As n is becoming 100, you can see the width of the peak has decreased. When n has become 1000, you can see it is such sharp peak is coming. That means n, capital N, which is the number of distribution planes. So as we are increasing n, which represents the number of distribution planes, your peaks are becoming sharper. The distance between the two peaks is also increasing, and the width of the peak is also decreasing. Fine. So that is what this question was talking about, that during column chromatography, as n increases, that is यहाँ पे capital N represents number of distribution planes. Okay, so as the number of distribution planes are increasing, the peak width also decreases. Peak width decreases and resolution increases. Okay, is it clear to everybody the concept? That there is a limit up to which you can keep on increasing the length of the column. You cannot keep on increasing the length of the column up to any extent. So therefore you are increasing the surface area of the column within the same length by having many number of distribution planes. Okay, so this is what is it clear to everybody? This concept is clear. Just say yes or no.
Ma'am, can you explain it again? I cannot understand.
So okay, so what portion you couldn't understand exactly?
Ma'am, about how the peak is goes up.
See, the basic logic is that we just said that the resolution, it depends on two things. Okay, it depends, it is directly proportional to the length and it is inversely proportional to the width. So that means when the length of the column is increased, when you are increasing the length of the column, when it increases, then your resolution also increases. Okay, but there is but you cannot, that means if you have a column of this length and if you have a column of this length, that means this column will have a better resolution as compared to this column, because it is a smaller length column. But you cannot keep on increasing the length of the column up to any extent. You cannot do that. Okay, so there is a certain limit up to which you can keep on increasing the length of the column. So what you can do is that basically what is happening when you are increasing the length of the column, the basic idea, the basic logic behind increasing the length of the column is that because the length is increased, and so it is getting distributed even more number of times. Because of the greater length, your mixture of arylite is getting distributed for a larger time here, to a larger extent as compared to this. So what I can do is that I can have some kinds of plates, theoretical plates. Now this theoretical plate concept I cannot explain you, because it is, then we have to go to really very deep about this chromatography. It will take even 1 2 hours, and we have to cover other subjects also. But anyways, if you want to know about it, you can read and you can ask me about this in the next class, subsequent lecture about theoretical plates. So what they did was, what scientists did was, that within the same column they have put different plates, many plates. So now these many plates are basically kind of increasing the surface area. Earlier, your mixture of analyte was getting separated only here. Now it is getting separated here, then here, then here, then here, then here, here, here. So it is getting more area to get separated. So the length, as the length of the column is increased, you have more area to separate the mixture of analytes. But you can also increase the surface area within the same length by introducing many number of plates. Distribution planes you can increase. And if I have distribution planes, just these are your distribution planes, 1 2 3 4. If I have distribution planes n is equals to 2, and I have distribution planes n is equals to 50, so n is equals to 15 means more distribution plates. More distribution plates means more surface area. More surface area would be better separation of the mixture. That means more would be the resolution. And obviously when the resolution is more, then the width of the peak would be less, because it is inversely proportional to the resolution. This is the basic general idea about how it is done. Okay. Now we will now we will move to the next. Will we discuss the questions? Yes. Now we will move to lecture 3, basics of chromatography. So here, in this chapter, we have studied about the chromatography system and the different forms of chromatography. Fine. So in very just quickly, we'll see that how the chromatography system works. So basically, you have a reservoir. So reservoir means reservoir of the mobile phases. This is the column. Okay. Now this column, what you can see, this very light grayish kind of thing is your matrix. That means your stationary phase is there in the column. Okay. And mobile phase you provide from outside. So this is the pump. This is actually the reservoir of the mobile phase. With the help of pump, your mobile phase comes and it goes to the column. So this is how your mobile phase is going to the column. So basically mobile phase is going to the column with the help of the pump. Now depending upon your experiment, depending upon what you want to purify, you can have one mobile phase or you can have a mixture of mobile phase. Suppose if I just want to use one mobile phase, then only this mobile phase will go and it will go inside the column. But if I want to use mixture of mobile phase, then here when these mobile phases, then it will be get mixed here and then it will be inserted into the column. It depends. Now this is your injection sample injection port. So just about above the column, if you have seen the video that sir has provided, then you will inject your sample from here, and then you will have peaks. You will have a detector where you will get the peaks, and then whatever fraction that you want to collect, that you can collect from here. So this is a basic gist of how a chromatography system works. So now we'll do the questions of this particular thing. Okay. Okay. First I'll, what is the next part? Okay, first we'll do the question also. So difference between partition and adsorption chromatography is, in adsorption chromatography, Molecules are not adsorbed on the matrix. In partition chromatography, molecules are not adsorbed on the matrix. Where is an adsorption? Molecules are absorbed. In adsorption chromatography, which matrix and analytes form mutual interaction. That does not happen in partition chromatography, both B and C. What is the answer? Try to answer the question. What can be the answer? Try.
Actually, these lectures were uploaded yesterday only, so I was not able to watch the lectures.
Okay. Okay, anyways, I'll just explain you quickly about what is partition and adsorption chromatography. Fine. Basically, adsorption. So, do you know the difference between absorption and adsorption? So, there are two terms. First is absorption, and the second one is adsorption. Okay. So absorption means that something is getting absorbed, means something is getting inside the surface. And adsorption means that it just sticks on the surface, it does not go inside it. So suppose uh, nutrients, so nutrients, cells absorbs the nutrients. Okay, cells do not adsorb the nutrients, cells absorb, because cells take up the nutrients inside it. So in adsorption, if this is a surface, and if molecules are just sitting on the surface and they are not going inside the surface, then that is known as adsorption. And if the molecules are going inside the surface also, then this is known as absorption. So is absorption and adsorption clear now? So what is the difference between partition chromatography and adsorption chromatography is that whatever matrix you have, so what, see, this is your analyte. Analyte means mixture which you want to separate. So when your column, in your column, this is your matrix, and when you are giving the mixture, then the molecules, they will form a mutual interaction with the matrix. The interaction can be hydrophobic interaction, ion exchange interaction, or whatever kind of interaction it will form, and then other things will be eluted out. Whereas in the case of partition chromatography, the uh, interaction is not happening. I'll just explain you like this. Okay. So, and second is adsorption. Extremely sorry for my very bad handwriting, but I'm very new to this using. Okay, anyways. So, in partition chromatography, this is your column. This is your column. Okay. Inside the column, so how, what are the two phases that we have in column chromatography? Name the two phases that we have in column chromatography.
Mobile phase and stationary phase.
Yes, mobile phase and stationary phase. So which phase is present inside the column?
Stationary phase.
Stationary phase. So stationary phase. So stationary phase can be in the form of a matrix. That means chem, some kind of functional groups or anything which is embedded on the column as a matrix. It is in the form of a matrix. Okay. So this matrix is nothing but a stationary phase. Fine. This is your stationary phase matrix. This is your stationary phase matrix. Okay. Now, this is your mobile phase. This is your mobile phase. And this is the mixture of analytes that you want to separate. I am just naming it as A. Now, in the case of analytes, what happened? Suppose this analyte have two things, A and B. Okay. Now, this B has the affinity for the matrix. So, B will bind to the matrix. Okay. B will bind to the matrix. But A does not has the ability. So, A will just come out. A will come out. So, this B is bind to the matrix. This binding is actually your adsorption. Why it is called as absorption? Because B is not going inside the matrix. It is just bounded to the surface of the matrix. Okay. And this, how it is binded? How it gets bind to the matrix? Due to various kinds of interactions. That can be ionic interactions, hydrophobic interactions, or different kind of interactions. So this is known as adsorption chromatography. And the other kind of chromatography is partition chromatography. Now, uh, just a minute. Hmm. Another thing is partition chromatography. Now, in partition chromatography, the stationary phase is also liquid. Okay. This is your stationary phase, which is also liquid. In partition, your stationary phase is also liquid. And when you put your mobile phase is also liquid, and when you put your molecules, then the molecules distribute between the two phases. So suppose if you have a stationary phase of oil, okay, suppose if you have a stationary phase of oil, just a minute. If I talk about partition, the stationary phase is also a liquid, and mobile phase is also a liquid, and whatever the mixture of analyte is there, it distributes itself between the two phases, but it is not binding, obviously, because there is no solid stationary phase. So you have to understand that stationary phase can be a solid also, and it can be liquid also. Whatever techniques that you are going to study now, in those your stationary phase is a solid, but stationary phase can be a liquid also. So when your stationary phase is liquid, then the separation is only depending upon the partitioning of the mixture between the two phases. Suppose if you have stationary phase as oil and mobile phase as water, so you know that the oil, the properties of oil are so different from the properties of water. And if you put a polar this thing, and if you have a mixture which has polar molecules as well as non-polar molecules, okay, so what will happen? Because your stationary phase is oil, so all the non-polar molecules, they will get partition into the oil, and all the polar molecules will be come into the water, and when that water will come out, all the polar molecules will come out. This is your partition. So partitioning means, in short words, how you can differentiate between partition chromatography and adsorption chromatography. In partition chromatography, your stationary phase is also liquid, whereas in adsorption chromatography, your stationary phase is not liquid, it is a matrix. Is it clear? Is it clear now? Are you able to get the
Yes, ma'am.
difference between
Yes,
partition and
ma'am.
adsorption chromatography?
Yes, ma'am.
Okay, if you have any confusion, you can ask. So now, so this is what we know. Now we know that in adsorption chromatography, when we talk about adsorption chromatography, so we said that the stationary phase is also solid, and suppose if B is getting bound with the stationary phase, there is some kind of interaction here. Okay. Now this interaction can be ion based interaction, It can be a hydrophobic interaction, or general this B has some kind of affinity to bind to this matrix. So based on the different kind of interactions that can happen between the molecule and the stationary phase, we have different kind of chromatography techniques. If the interaction is ionic, then that particular chromatography is known as ion exchange chromatography. If the interaction is hydrophobic, then that kind of interaction is known as hydrophobic interaction chromatography. If that interaction is because B has an affinity towards the matrix, then that is known as affinity chromatography. So whatever chromatographic techniques that you are going to study in this particular lecture, they are actually the different varieties of adsorption chromatography only. Is it clear? Is it now clear? I hope it is
Yes,
clear
ma'am.
now. Okay. Now we'll do the question. Now you read the question number 9 and try to answer this. Tell what is the difference between partition and adsorption?
Ma'am, D.
D. Okay, D is in partition chromatography, molecules are partition. Okay. Yes.
Both B and
So the
C.
answer is D. Both B and C. The next question is, in low pressure chromatography, the types of pump used are peristaltic pumps, syringe pumps, infusion pumps, and steel pumps. So in low pressure chromatography, basically the kind of pumps that are used are peristaltic pumps. Now next question is, the analyte is injected into the chromatography system by which means? Through the column which contains the matrix. Analyte is mixed with the buffers and added to the column. Through a syringe that loads the sample into the column, or through a peristaltic pump.
D.
Ma'am, D.
Through a peristaltic pump, no.
B.
No, B is also not the correct answer.
C.
Yeah, C is the correct answer. So basically, uh, just a minute, just a minute. Mm, you see here, so sample is injected with the help of a sample injection loop. This is a sample injection loop. So with the help of the syringe, you load the sample with the help of the sample injection loop. So basically the loading of the sample occurs with the help of a syringe. Through the, so there is a port which is known as sample injection loop, where you can just with the help of a syringe you can load the sample. Um, okay. Then we'll come to lecture 4, which is ion exchange chromatography. So uh, so we have studied about partition chromatography and adsorption chromatography, and we have studied that in partition chromatography, basically the stationary phase is also mobile, and the separation occurs because the analyte, the mixture of analyte, it gets separated between the two phases. Whereas when we talk about adsorption chromatography, the stationary phase is basically a kind of a matrix which is also uh, solid, and the partition occurs because certain molecules, they get attached, they get adsorbed on the matrix because of some kind of interactions that happens, and other molecules that do not, that could not form interaction with the matrix, they are eluted out. So now in accordance to with, we will first start with our first kind of ion exchange adsorption chromatography, which is ion exchange chromatography. I think someone has messaged me. Just a minute. Okay, someone wants to join. So for this, first we'll see. Just 1 minute. Hmm. Hmm. Fine. So basically, if we talk about a protein, so the different kinds of adsorption chromatography that has been developed, the ion exchange chromatography, affinity chromatography, size exclusion chromatography, so these all have been developed by exploiting the different properties of a protein. So if you see that proteins can have different charges. Okay, so protein can have different charges on them. Some proteins are positively charged, some proteins are negatively charged, some proteins are uncharged. Even if you talk about two positively charged proteins, the distribution of charges can be different. Some proteins can have more number of positive charges, whereas some proteins can have less number of positive charges. So the proteins can have difference in the charge, and the charge difference, it depends upon the functional group. So every property of protein, the distinguish properties of protein, or the difference in the property of protein, is because of the functional groups which are attached to the proteins. The functional groups carry the charges. The functional group determines the affinity of the protein to other molecules. So first thing is charge. Second thing is hydrophobicity. So hydrophobicity means that this is a protein. So protein will have hydrophobic, proteins have hydrophobic amino acids also, and proteins have hydrophilic amino acids also. Someone is prompting me, just a minute. Okay, someone wants to join. Fine. So proteins can have hydrophobic amino acids also, and proteins have hydrophilic amino acids. Now, when a protein is in water or in a polar solvent, what happens that the hydrophobic amino acids, so the protein folds in such a way that the hydrophobic amino acids are in the interior, and exterior whatever amino acids are there, they are hydrophilic. So hydrophilic means water loving, and hydrophobic, phobia, phobia. So I have a phobia of water, that means I'm scared of water. So hydrophobic means they are water repelling. So whatever hydrophobic amino acids are there, they're inside, and hydrophilic amino acids, they are on the outer side. So what this hydrophobicity is exploited. So what is done is that these hydrophobic amino acids, they interact with the matrix, and then you can separate the proteins. So this is your hydrophobicity. Second is the surface area. Surface area means some proteins have functional groups which are very large. So proteins will be very large, or some proteins have 10 functional groups attached to them. Some proteins will have just two functional groups attached to them. So protein which has 10 functional group attached with them, it will have a greater surface area, whereas proteins which have two functional groups attached to them will have a lesser surface area. So this surface area property is also exploited to separate the proteins. Then comes your affinity. So affinity means there are two proteins X and Y, and there is a molecule matrix which is A. This X has very great affinity to A, whereas Y does not have any affinity to A. So certain proteins have the characteristic affinity to certain molecules, and that affinity can also be exploited to separate the protein. So there are different kind of adsorption chromatography, and all the different kind of adsorption chromatography are based by exploiting the different properties of proteins. It can be charge, it can be surface area, it can be hydrophobicity, it can be affinity. Okay. So, so if you see, by taking the property of charge, you have ion exchange chromatography. So ion exchange chromatography is a kind of chromatography where you can separate the positively charged or negatively charged proteins. So proteins which are charged, They can be separated with the help of ion exchange chromatography. Hydrophobic interaction chromatography means by taking out the hydrophobic inner shell of the protein, you can separate those proteins with the help of HIC. Hydrophobic interaction, we call it in short as HIC. Gel filtration chromatography means of surface area affinity chromatography by exploiting the affinity. This I will explain in the next class when you will study about it. So, depending on this, let's solve one or two questions. Okay, so match the following. Now try to match and tell the answer. I have explained you this. Try to answer.
Ma'am, A with the hydrophobicity.
Hydrophobic interaction chromatography with hydrophobicity. Yes. Ion exchange chromatography depends on which one of this?
Charge distribution.
Yes. Charge distribution. Gel filtration chromatography.
Surface area.
Yes. Surface area. And affinity chromatography with different ability to bind to certain molecules. So this gel filtration chromatography we will be studying, I think, in your next week class. So I just had one question. So this is like today is Tuesday. So when you have to submit your assignments, what is the last date for you to submit your assignments? When you have to submit your assignments? Like weekly assignments.
Ma'am, today is the last date for submitting sixth assignment.
Submitting sixth. Acha, okay. Okay. So that
Before
means
12 p.m.
so this week's assignment you have to submit next Tuesday. It's like this.
I don't know. Don't have any
Don't know?
idea, ma'am.
Are you not
I'm,
submitting the assignments?
I'm submitting the assignment, but I have not clear idea. But I am doing the assignments.
And when
It will
your
be
lectures
submitted
are
in next
uploaded,
Tuesday.
See, I saw some of them.
matlab
Yes,
on
ma'am.
weekly
This
basis, is
assignment
there
is
any
submitted in next week,
what? Can
Tuesday.
you be a little louder?
Yes,
This
ma'am.
assignment is submitted in next week.
Okay, okay. And when your lectures are uploaded, week on weekly basis, when your lectures are uploaded, any specific day when your lectures are uploaded?
Mostly Monday lectures come.
Okay, so Monday lectures come and then Tuesday you have live class. Fine. Anyways, so now that we know that the different properties of proteins are exploited for different kinds of chromatography, first we will go through ion exchange chromatography. So ion exchange chromatography means that if a protein molecule has a certain charge on it, suppose a protein molecule is positively charged or negatively charged, then what we can do is, suppose we are here learning about ion exchange chromatography. Fine. Okay. So if this is my, these are my two columns. Okay. And this is my matrix. My matrix has molecules which are positively charged. And this matrix has molecules which are negatively charged. Okay, so if I add a mixture of analyte, it is also charged. So what will happen here? So suppose my mixture of analyte has positively charged molecules also and negatively charged molecule also. A is positively charged and B is negatively charged. Okay, here also A is positively charged and B is negatively charged. So what will happen? Here what will bind? In this matrix what will bind? A or B?
B.
B. So here B will bind. So whatever gets bind to the matrix, that is eluted at the last. So here what would be sorry. So here what would be eluted first? A. Because it does not get bind, it is bind, so it is not getting eluted here. Here A will get eluted. Here what will bind? These are negatives. So what will bind here? Tell, what will bind here?
A.
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 A will bind here. So what would be eluted first? Sorry, A. What will be eluted first? What will come down through the column
B.
first?
B.
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 Okay. So this exchanger, so this is what ion exchange chromatography is. That you have a cation exchanger or an anion exchanger. That means your matrix is either positively charged or negatively charged. And then depending upon the charge that is present in your this thing mixture of proteins, you can separate it. Now there is a question that okay, I have eluted A, but my protein of interest was B, but it is already bound with the matrix. Now how to isolate B? So what do you think, what can be the approach by how we can isolate B from here? How can we isolate B from the matrix? Because it is already bound to the matrix. That means we have to do something that can break the interaction between the positively charged matrix and the negatively charged B protein molecules. So what do you think we can do by which we can isolate B, elute B from the column?
By changing the pH of solution.
Okay, we can do the change. We can change the pH. So by changing the pH, ultimately what is happening? You're right. We can change the pH. So ultimately, what is happening by changing the pH? So whenever you change the pH, what happens actually in general? The charge distribution changes, right? So uh, the way by which we can elute B is, suppose I add another solvent which is C. Okay, this B has single negative charge, but this C has double negative charge. Okay, so now what will happen? This C will have higher affinity to bind to positive because it is even more negative charged. So there would be competitive binding. So what will happen, that as I will start adding C to it, this B will get this B will start getting replaced, and instead of B, this C will get bind because it is even more negatively charged. So this is the way how you can elute your sample of interest which is bounded to the matrix. So you can give a molecule which is even more charged than the molecule which is attached to the matrix. So what will happen? There will be competitive binding, and because the molecule, the solvent that you are giving is even more charged, either positive or negative, then this will be replaced by this C. Is it clear to everybody
Yes,
how
ma'am.
the illusion is happening? Okay. Now, so like this you can elute. Now, whenever we talk about ion exchange chromatography, we talk about, there are, see here you can see that either we can have a matrix of this type or we can have a matrix of this type. Therefore, there are two type of exchanger. So whenever we talk about ion exchange chromatography, we actually have two types of exchanger. We have a cationic exchanger and we have an anionic exchanger. So, what do you think? What would be a cationic exchanger? How would be the matrix of a cationic exchanger? And how would be the matrix of an anionic exchanger? What should I write over here? Plus or minus?
Ma'am, in
Cationic?
cationic exchanger, there is a plus charge.
No. Cationic exchanger will have a
Cation
minus
exchanger are
charge.
negatively charged.
Cationic exchanger will have a minus charge. Anionic exchanger will have a plus charge. Now, can anybody explain why? Cationic exchanger it should have a plus charge. Why I have
Cat-
written minus here?
Because
Then
cations are positively charged, so that will exchange by the negative charged molecules. So cationic exchangers are negatively charged and
exactly,
anions are positively charged, so they will be replaced by negatively charged molecules.
exactly, very good. So when I talk about cationic exchanger, you have to understand it's very important. Again, very common question in many competitive examination. It's asked a lot because people get confused. They think that cationic exchanger, okay, it will have positive charge, but no, cationic exchanger has negative charge. Why? Because cationic exchanger means it is exchanging cations. So to exchange cations, the matrix has to be negatively charged, then only it will be able to exchange cations. Na? Cationic exchanger means that you are getting cations by the help of this matrix. So if you want cations from this matrix, you have to have a matrix which is negatively charged. Similarly, anionic exchanger means that what you are getting are anions. It is exchanging anions. So to exchange anions, to get anions, you have to have a matrix which is positively charged, which can attract cations. So how to remember? Cationic exchanger means it attracts cations. Okay, simple technique to remember. Cationic exchanger, it attracts cations. An anionic exchanger, it attracts anions. So to attract cations, it itself has to be negatively charged. To attract cations, it itself has to be, so to attract anions, it itself has to be positively charged. Is it clear to everybody?
Yes, ma'am.
Yes, anybody was saying something? Some girl? Any problem? Okay, fine. Okay, fine. So this is important. Cationic exchanger and anionic exchanger. So there are different kinds of cationic exchanger and there are different kinds of anionic exchanger. So you can see these, the list that is given over here. These are some of the very commonly and very important. This list is important. Try to learn this not just for your this certificate point of view, but in general for your competitive examination point of view also. So this DEAE, this is very important. These two are also very important. So try to learn this. So in this lecture we have studied about, you can see here, cation exchanger means it will, so this is your matrix. In cation exchange, the matrix is negatively charged. In anion exchange, the matrix is positively charged. Okay, so it can attract the cations, it can attract the anions. So this is your cation exchanger and so likewise, so your ion exchange chromatography can be of two types. First is cation exchange chromatography and the other one is anion exchange chromatography. Is it clear? Now we'll solve the questions. Just a minute. These questions were done. Okay, first we have to deal with this question. So in cation exchange chromatography, the matrix has, tell me what is the answer?
Negative charge.
The matrix has negative charge. Yes, right. Then, actually there I have done so much of it's not very tidy. Now tell, identify the cation and anion exchanger respectively. So which one is the cation exchanger in all of this, and which one is the anion exchanger in all of this? Tell anybody who remembers.
Carboxyl methyl is cation exchanger. DEAE is anion exchanger.
DEAE is anion. So which option you think is the right one here?
Option B.
Option B. Correct. Then we come to 15th question, which is for below and above pi value, select the type of exchanger to be used.
Cation and anion respectively.
But we have a lot of questions. Okay, so below is cation and above is anion. Yes, right. So there is a general. If you have read that whenever we talk about proteins and the pH, so if the pH is less than pi, then what charge do we impart? We impart a positive charge. If pH is greater than pi, then we impart a negative charge. This is thumb rule. You have to remember. Okay. Then we come to the next class, which is the last class, hydrophobic interaction chromatography. Okay, so in hydrophobic interaction chromatography, just a minute. So before this, this ion exchange chromatography has a lot of applications. So this ion exchange chromatography can be used for studying the protein DNA, for studying the protein DNA interactions. Then this ion exchange chromatography can also be used for studying the kinase assay. This you have to just go through it. They are just the applicatory part of ion exchange chromatography. Then it can also be used for the softening of the water. Okay, so now we have studied about ion exchange chromatography. So we were studying about adsorption chromatography. Okay, so in adsorption chromatography, depending upon the different properties of the proteins, there can be different kinds of chromatographic techniques. The first one is depending upon the charge. So depending on the charge, we have studied about ion exchange chromatography. Now ion exchange chromatography can be of two types: cation exchange chromatography and anion exchange chromatography. In cation exchange chromatography, the matrix is negatively charged. In anion exchange chromatography, the matrix is positively charged. Okay, this we have studied. Now, next technique that we are going, next type of chromatography, under these are all adsorption chromatography techniques only. Okay, we are not studying about the partition chromatography. This is under adsorption. So next technique that we are going to study is hydrophobic interaction chromatography, which exploits the property of hydrophobicity of proteins. Okay. Now what is hydrophobicity of proteins? See, you have to read about this. I mean, you have to, if though for those students who have not read the studied the lectures, I would suggest to go through the chapter, especially this one, because sir has very beautifully explained about the hydration cell, the hydration shell, and the hydration layer, and how the hydration layer is removed, and how salting in and salting out happens. I will tell you in a very brief about it, so that just for the sake of your basic understanding. So basically, as I told you, that suppose this is a protein, right? This is a protein. So protein, or each and every protein has a different conformation in which it gets conformation in which it gets fold. So every protein has a defined folding structure. every protein has a defined folding structure. And whenever we say that denatured the protein is denatured. So what is the meaning of denaturing of protein? Denaturing of protein means that the three-dimensional structure of protein is, or the quaternary structure of protein is lost, or it is damaged. So the protein is folded in such a way that the hydrophobic amino acids, they are present in the interior, whereas the hydrophilic amino acids are present out the outer. So that when this protein is present in a solvent, in a polar solvent such as water, these hydrophobic amino acids are protected. So what happens actually is that water molecules, they are present like this. Okay. So like this, water molecules are present all over the protein. And these water molecules, and this is the hydrophilic area, so they can interact with no problem. So this kind of forms a hydration shell. That means, so the protein has a kind of a shell of water molecules which is known as hydration shell. So hydrophobic, when we talk about hydrophobic interaction chromatography, so we exploit the hydrophobic amino acids. So what is actually done in hydrophobic interaction chromatography is that you have a column and you have a matrix. So this matrix has affinity towards the hydrophobic amino acids. So the hydrophobic amino acids, they will interact with the matrix, and like this it happens. Now, we know that the hydrophobic amino acids are present in the interior. That means something has to be done so that these hydrophobic amino acids are exposed out. Okay, we have to expose these hydrophobic amino acids so that they can interact with the matrix because they are present inside. So, the first step of exposing these hydrophobic amino acids is that we have to break this hydration shell. Once this hydration shell is broken, then it would be easier for us to take out these hydrophobic amino acids. Okay, so for breaking out the hydration shell, what we do is that we add salt. So there are two things, there are two terms. First term is salting in, and another term is salting out. Again, very important. Students, this lecture, this whole seventh lecture is very important in terms of your any competitive examinations, very especially your GATE and DBT. Because in GATE and DBT, questions related to chromatography, questions related to salting in, salting out, the different numerical problems related to chromatography, they are very frequently asked. So that is why I'm saying that whatever you are reading here and whatever you will be reading in such lecture will try to make your basic concepts that will be very useful for understanding the integrate details of these particular techniques. So even you can, even who have not, who could not go through the lectures, you can go through the lectures, and in the next subsequent class also, if you have any problems related to these techniques or otherwise, you can ask me, because this is the kind of basic concept building, right? Now, so we are talking about salting in and salting out. So what is salting in? Addition of low amount of salt to the protein results in the displacement of bonded water molecules with an increased protein solubility. This effect is called as salting in. So what happens is that salt, when you are adding salt, so the hydration shell kind of gets broken. So if you want to break the hydration shell, what you have to do is that you have to add salt. So initially when you are adding salt, what is happening? The solubility of that protein is getting increased. But after a level, what happens? That there is a saturation level, that now no more salt can be taken up by the protein. And after that, the solubility starts decreasing and the protein gets precipitated. So that point when you're adding that particular amount of salt, that the protein is getting precipitated, that is known as salting out, and that causes the precipitation of protein. So salting out, a general statement which is very important, salting out causes the precipitation of proteins. Okay, not salting in. Salting in will increase the solubility as you're adding. So if you have a protein and you start adding salt, so what happens, that because you are as adding salt, so the water molecules are getting displaced, and because the water molecules are getting displaced, you have even more area to bind. So initially the protein solubility will increase, but as you start adding more and more and more amount of salt, what happens, that not only the water molecules are getting displaced, but also the hydrophilic patches of the proteins, they also start getting displaced. And because the hydrophilic patches also starts getting displaced, then your hydrophobic patches are exposed out, and because your hydrophobic patches are exposed out, then your protein becomes very much insoluble because it is present in water, and then it gets precipitated. Is it clear to everybody? Salting in and salting out, is it clear?
Yes, ma'am.
Yes, ma'am.
Okay. So, thumb rule, when you are adding salt, solubility is increasing. You keep on adding salt. First, your hydration shell is removed. Okay. Protein is very happy that my solubility is increasing. But as you are increasing more and more and more salt, the hydrophilic amino acids are also getting released, and then you are left with the exposed hydrophobic patches, and that will cause the precipitation. That is your salting out. Now we'll come to the questions of this particular this thing. First question is, protein DNA interaction can be studied by, quickly, how it
Ion
can be
exchange
studied?
chromatography.
Ion exchange
MC.
chromatography, yes. Precipitation of proteins during HIC, that means hydrophobic interaction chromatography, can be achieved by salting in,
Salting out.
salting out, changing the pH. What
Salting
is the
out.
answer?
Salting out.
Salting out. Correct. Okay, just are you able to read this? For equilibration of HIC matrix, the most commonly salt used is? Does anybody know?
Ammonium sulfate.
Yes, good. Ammonium sulfate. So again, one thumb rule, for salting out, we have to add some salt. So the most commonly salt that is added for salting out is ammonium sulfate. So for salting out, which salt is added? Used ammonium sulfate. You can use KCl and NaCl also. Okay, any salt can be used. Even you can use KCl and NaCl. But the problem with KCl and NaCl is that for achieving the same level of precipitation, you have to add even more amount of salt, and that could hamper with your protein integrity and your purification. That is why you add ammonium sulfate. Next question is, what is used for the regeneration of column after HIC? So that means that this is your column, okay, and this is the matrix, fine, and your hydrophobic molecules have bounded to it. Now for eluting these and for the regeneration of column, so that the column can be used again, so this thing also you should remember, that it is not like that you can use the column only once you use the column and you discard it. No, you can use the column many number of times. So for that you have to treat it. First you will elute your sample of interest, and after that you will treat your column in such a way, you will clean it, you will wash it several times, so that nothing is stick to the matrix, and then you can use the column again. So for the regeneration of column, so that it can be used again, what you can use, tell me? 6 molar urea, guanidinium hydrochloride, ammonium sulfate, both A and B, or both B and C? Anybody knows the answer?
Ma'am, both B and C.
No, the answer is both A and B. So either we can use 6 molar urea and we can use gonadium hydrochloride for the regeneration of coral. So the answer is both A and B. Fine. Next question is, ion exchange chromatography is based on partition chromatography, electrical mobility of ionic species, electrostatic interaction, adsorption chromatography.
Adsorption chromatography.
Okay, ion chromatography is based on adsorption chromatography. That can also be correct. But amongst these, what you can
B.
think is the most appropriate answer? Ion exchange chromatography is based on adsorption chromatography only. But more specifically, like in adsorption chromatography, if we try to even narrow down it, more
Ma'am,
specifically,
C.
it is based on the attraction, electrostatic attraction. Yes. Then question number 21 is separation of proteins based.
Adsorption is one type of ion exchange chromatography.
Sorry, can you repeat your question? Ion exchange chromatography is a kind of adsorption chromatography. Okay. Okay. Next question is separation of protein based on their size and shape is performed by gel filtration, ion exchange, affinity, adsorption. Based on their size and shape.
Adsorption chromatography.
See, you have to answer more specifically. So, yeah, separation of proteins based on their size and shape. Ultimately, it's the kind of adsorption chromatography only. But more specifically, it is the gel filtration chromatography. So, I think we will be studying about gel filtration chromatography in our subsequent this thing lecture. So, based on the gel filtration means you have a lot of beads here, and depending on the size of the protein, if the size of the protein is uh, even bigger than these beads, then they will not go inside, it will just come out. And if the size of the protein is smaller, then it can go inside. So I mean, you will study when we will study about gel filtration. But as of now, just remember that separation of proteins based on size and shape is performed by gel filtration chromatography. It is also known as size exclusion chromatography. Okay. Next question. Next is cell disruption and release of products in a bead mill follows first order, second order, zero order, pseudo first order. So, what do you think is the answer of this question? The answer is first order kinetics. You have to read about it. Anyways, second question is affinity chromatography separation is based on duration of analyte, specific interaction between analyte and ligand, molecular weight or flow through time of the analyte. What is the answer?
Molecular
B.
weight.
No, molecular weight is not the
Specific
correct
interaction
answer.
between analyte and ligand.
Yes. So you have to understand affinity chromatography means affinity means what? What is the affinity of the analyte with the matrix? So basically in affinity chromatography, this is your column and this is your matrix. So the matrix has some kinds of molecules attached, and the uh, analytes that you are providing, that you want to separate, some of them have affinity towards those molecules, and those are actually the ligands. So therefore the affinity chromatography separation is based on specific interaction between analyte and ligand. Okay, so I hope this was clear to you. So if you have any questions, any queries, you can just read this and we can discuss the queries in our next class. This is a very important topic, so you need to have a clear understanding because this will form the base for further even difficult questions. So if you see what kind of questions comes in the examinations, so the questions are not that easy, okay? They are a little difficult, they are a little tricky, they are mostly numerical based. Some of them are conceptual based, but there are many questions which are numerical based. So you have to have at least the basic knowledge, the basic idea of the chromatography, the basic principle. And then anyways, we can see and read about it more. If you have any queries, we can answer it also. Okay, so we'll stop the class now, and we'll meet in the next week. Bye bye.
Thank you. Happy Teachers Day.
Thank you.